Day 11. Welcome to the study, to the bench. We still don't have a lab code. As soon as it arrives, we will do experiments. But right now, going with the usual, just learning everything what we can, not in a linear fashion. That's important. This is not chapter by chapter learning biology and science. It's curiosity driven. We identify the gaps and using AI, of course. Using AI for everything. Okay, so here, last time we stopped when we asked Claude to create us a pipeline to verify the ideas. And we throw the first idea and so on. And it was working. Aggregate scored 15 out of 15. Fire stage none. Score shaped warning, mild. Raw idea, unusually self-aware, could be careful thinking or rubric awareness. Very interesting. So, I mean, what's wrong here anyway? If you remember last stream, we basically created the pipeline where we're gonna validate ideas. And the problem with the AI agents and so on, we discussed that they actually very
sycophantic.
So we're trying to minimize that as much as possible because we're doing science here. I mean, we are learning how to do science. So we created a couple of agents. I mean, when I say we, the Claude created agents for us, and to be super transparent, we did not even read them. No, we strictly vibe coded it. We give a prompt, we click accept button, and never verified the result. That's a classic definition of vibe code or vibe learn or whatever it is. But let's see what it says. How to behave. The hard constraint is discriminating prediction. If both the hypothesis and the leading alternative predict the same observation, the experiment cannot distinguish them, and it's not worth running. Not bad. I mean, that's as much as we're gonna read it. Like, it's so hard to even finish this. I don't know why, but it's so hard to finish reading this. If you cannot find the discrimination prediction, your output should say so plainly. That is itself the most useful things you can tell. Blah, 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 blah. Again, can't read this for... We will read that eventually, or maybe not. But, so we ended the stream yesterday. So day 10, we ended with a question. Is there... Where is my question, by the way? For 12 minutes. I don't know what's this line about. Oh, that's the agent? Is it? Very interesting. So the raw idea was that is there analogy between the classic, basically the idea was there from a childhood, from a 5th grade, when you first learn chemistry or physics, and you know that electrons run orbiting around the atom, and the sun we have, aka atom, and planets orbiting around sun, aka electrons. So, and we asking like, is there analogy in terms of ratio or proportion of the distance. Therefore, if the analogy that our observable universe or galaxy mimic the distance between molecular structures, and what I'm saying is wrong here is most likely our agents and pipeline that we white-coded it is wrong because it scored 15 out of 15. So if 15 means how bad it is, or how untrue or not viable it is, then we did a great job. Because it's 15 out of 15. It's idea from a 5th grade. When you just start thinking for the first time, you mature, you start growing hair as a man, and you start applying some analogy in your thinking. So if it's 15 out of 15, it's how good it is, our pipeline is useless. Failure stage, none. But let's read it, right? That's what we are here for. Pipeline atom galaxy analogy. 05 synthesis. Okay, we're gonna close this. Oh my god, this is a lot of text. It's already making my brain, ADHD brain, go nuts. To start reading this, it's easy. I just jump into it. But to finish reading this, for me, I don't know, it is for everyone this day and age because of swipe culture, right? But for me to finish this reading, that's probably as hard as to do 4 sets of squats with my max. I don't know. It's like going to the gym and doing workout. The hypothesis as it now stands. The original intuition that an atom and the solar system share some deep structural kinship contains one well-grounded piece, one already falsified piece, one category incoherence piece, and one unfalsifiable piece. They should be held separately. So far, I love it. Again, we're not judging an idea, right? Like, think about it. We're not judging an idea about it. We on purpose, we're doing a unit testing, right? This is a software development 101. You develop AKA software, you wipe coded it, a pipeline, let's say, and you feed it knowingly bad input or incorrect stuff. And the software should understand it and behave to reject it. That's what we're doing here. So this sentence saying that's actually doing the right things. So it's saying that it already has a pieces that couldn't be falsified, or one category by the way, this category, like, that's gonna be a sidetrack. I really like, and I think I already talked about it, but I'm gonna do it one more time. I really like that now in modern science we're going back into remove the borders between categories. And what I mean by that, not just remove borders from categories. Categories is still useful when you want to overglance something and just describe things quickly using not a lot of sentences, right, or words. So categories as good as a compression mechanism to convey the idea. But right now, specifically when we're talking about stuff like biotech, it always in a border between chemistry, physics, and biology. And we should remove a category. And I like that trend in modern science that we are removing those categories. That's when we go deeper into the question why it happened. On an organic level, we go deeper now that we start asking a chemistry question, or why this reaction is happening on a molecular level. And then we start asking a physics question like, or why actually electrons doesn't bound to this molecule, right? So, and this is so cool because categories are always abstraction. Like, where do you draw the line? What I really like that's from Michael Levin, he advocating that we should remove those strict borders between categories and look at everything as a spectrum. And that's what I really like. Like if we open a spectrum of color, for example, right? Or light, like take a look at this. Yeah, maybe. I just want to go full screen on it. So where's the border, right? Like, how are you going to draw a border? Where's the green, for example, ends and starts? Oh, so is it ends here, or right here, or this one? So this is green and this is yellow, or this is yellow, this is green. Like it's so. Oh my god! I don't know why there's flies here. So cool. Close it. And that's what we mean by like removing categories. It should not be a category like this is green, this is yellow, this is red. Yes, it's useful in the context when we're saying like, hey, the traffic light was red and not green. You know what that mean, right? But when we look into this and start arguing, like when we zoom in so dramatically, that we start arguing like, oh, this is green or this is yellow and so on, of moving that line, it's useless. We should remove those lines and look at this as a spectrum. And this is, as Michael Levin was given another example, like in biology, as the organisms grow, as we grow from a child to adult to, I don't know, the next category even, elder person, like 18 is not a border, right? Like 18, you, or 21, you can like start drinking. Like it's not a, like, yes, it is arbitrary border, but we should not judge person differently if he or she a little bit left or right from that border. That border and categorization strictly useful in certain cases. But we should look at everything as a spectrum. That was a tangent side quest. So one category incoherent space, this should be like, I'm not sure, like, I love spectrums. And by the way, I'm judging all this work and millions of tokens, a real physical dollars that's went into this research, if we can call it research, just by first sentence. So let's see if we can finish this. This is actually a very interesting challenge. Can you finish 3 pages of reading? The well-grounded piece claimed to the two-body inverse square force problem produce mathematically identical classical orbiter solution regardless of whether the force is gravitational or electromagnetic. Oh, this is interesting. Okay. This is not speculative. It is established classical mechanics captured in Bertrand's theorem and the SO4 symmetry of Kepler problem. Kepler problem. And the hydrogen atom. Interesting. I want to see what Kepler problem is. Kepler is a special case of two-body problem. I mean, right away, the TV show Three-Body Problem, that's how brain jump into like different topics. And we open a new tab for Netflix and we start watching, is it significant accurately or not. Two bodies interact by its central force that varies in strengths. Okay, I got the idea. Go back to the article. And the hydrogen, the atom solar system visually analogy reflects the real mathematical equivalence, but only in the two-body. Classical mechanics limits. This is the legitimate core of the original intuition. Okay. Claim 1. The already falsified piece. Claim 1. The ratio of electron orbital radius to proton radius in hydrogen ground state is approximately 62,000. The ratio of Earth's orbital distance to the Sun radius is approximately 215. This differs by a factor of roughly 290. The ratio of hydrogen ground state is approximately. So you're saying is, let me understand that correctly. The ratio of the size of the electron of the atom to the distance between of the radius of the orbit is 62,000 versus the Sun and first planet 290. The specific structural ratio that was supposed to be preserved across scales is not preserved. This is calculable directly from standard constant and has been confirmed by a textbook problem. The ratios. Oh, like I should tell my agents to speak basic English when they create the synthesis, because I don't know those words. Ratio. That should be a ratio, not a. listening, not by reading. And that created a huge artifact for me. Or crutches, I would say, right? A limiting factor. I'll tell you a story. So at some point when I was learning English as I moved to the United States, it was a time when if you give me a list of paper with a written text on it and ask me what is written there, I would try to read it and I maybe will get like 20% of what it is. Like it would be so hard for me to read it properly or at all read it and comprehend, but at the end I will get only 20% what's in the paper. But if anyone, and I mean pretty much anyone with any accent, not with any accent, but if classic American accent or American reads same text to me, I will understand 100% of it. And I'm sure it's not just me, it's like pretty common when you learn foreign language. But I got into that local minima. And what happened, I got so comfortable. So it was uncomfortable for me to kind of unlearn and go and study grammar and read more and like study properly. Properly, I mean like by looking at the word and how they pronounce, pronunciation of the word. So I basically developed these crutches for myself that I see the word that I never seen before. For some reason, I don't know why I didn't see that word in writing. And I read it as a ratios, and I don't know what it is. But the word
Ratios.
ratios, of course I know, right? It's everywhere. And my brain did not associate this with the ratios.
Ratios.
Anyway, another sidetrack. So by the way, it's time to count how many sidetracks we have. And that's, my friends, another sidetrack. The idea of count sidetracks or side quest is on itself a side quest. So color, we're gonna add, we're gonna add capture browser color. We want text for type, side quest. But doing the scientific stuff, we're gonna see the definition of the side quest. And is it related to attention deficit order? By the way, I love that you can misspell stuff to Google and just figure out. Side quest is an optional, minor, or unexpected task that deviates from a primary goal. Side quests. Okay, the text would be side quest count, CQC. Right now we have 2, now the 3. And word is. We'll make it red, right? Select color. Red. 2 color, outline, drop shadow. Select color. Red but black. Spectrums. CQC. Okay, 3 side quests so far. Not bad. classical mechanical legs such as gravity, A, V, R minus everything. Oh, this is so tempting. This is so tempting to click on it and go all in. The ratio can be made to match by choosing a sufficiently large quantum number n, but the n has to be chosen specifically to produce the match, which means nothing is being predicted. Let's collaborate on this claim number 1, already falsified piece. So explain me in basic English, not scientific one. What ratio actually between the size of the, let's say, sun and the planets around it and the distance versus the size of atom and electron and the distance between them? And yes, we do know that the forces that keep electrons around the atom are different than the forces that keep planets around the sun. But let's pretend those forces are not the point, and we just want to see the ratio of size compared to the distance. In terms of what exactly we're going to take as example, is it our solar system or different suns and different planets? That's I don't know. And also in terms of what element we're going to take in terms of atom and electrons, I don't know neither. But let's maybe put it this way. Let's create a normal distribution of what galaxies or star system with the planets around them actually match the molecular structure of, in a ratio, of course, the distance between atom and electrons. Also, let me know if I'm wrong, but the atoms are different size based on the elements, right? So they grow in size from hydrogen and higher. But electrons are the same, pretty much, right? And how that match the different galaxy and star systems? We do know that sun could be different sizes, very different, but other planets around it more or less the same. Okay, I give the unrealistic task to AI. It's close to 4.7. Let's see how it goes. It's totally gibberish, but has some kind of like glimpse of maybe a right direction, or maybe something, I would say, a glimpse of hope, but it's definitely not comparable system because we don't have same scale to compare, unless we invent our own scale, or AI, that's what I'm very interested in, is invent its own scale. And yes, I know we should not play with the AI and see if it can invent something in terms of LLM and in terms of under like all these corporate harnesses. Of course, if we get a real open source with open weights LLM are like not LLM, probably even better, but LLM, we can be a little bit more in a novel directory, right? So it doesn't have all those harnesses of like be polite, don't say bad things, don't criticize, and so on. It could be more realistic and open head. By the way, that's what we're gonna do. Yeah, clip that. The clip that, the keyword, it's for content machine, so it can basically do highlights later on. I just need to get used to say that. We definitely should use the open source models, and so we can remove all these unnecessary filters, because those filters only will, like, it will keep forcing us into generic distribution, and we're not looking for something generic here. This is regard. By the way, the topic of this stream completely shifted from biology into pipeline and science, but it's good. We're testing our pipeline. I think I have an allergy, like I'm sneezing and coughing every 15 minutes. One month. Yeah, I think I have an allergy for sure. I have to put on mute every few minutes. A category incoherence piece, claim number 3. The claim is that splitting the sun produce remnants analogous to protons, charge energy producing, and neutrons, neutral, inert, does not survive contact with the actual physics of stellar remnants. Damn, are they like? Is a scientist really use all these words?
Remnants. Remnants.
Remnants. That's so cool. Neutron stars are classified by mass, density, and pressure support mechanism, not by electric charge. Yeah, we knew that. Stellar collision. And that's good. It's found in this. Stellar collision outcomes include black holes. Oh, interesting. Yeah, that's very good. Hypermassive neutron stars, kilonova, and gamma ray bursts, distributed continuously as a function of progenitor mass, not split into two clean categories. The proton-neutron distinction is a quantum chromodynamic property. Stellar remnants classification is general relativistic and nuclear physics property. There is no parallel classification system. Very good. Very good job. AI did a good job. It didn't fall into like tempting pitfall. What we got here? Oh, we had the answer here regarding the sizes, right? Still roughly the proton width. Electron sits roughly 63,000 proton widths away. Right. And solar system, sun, Earth, sun, radius, kilometer, Earth orbit, so it's 250. So they're way, way farther. Therefore, what comes, that we already know, the gravity is way, way weaker than strong nuclear force or the weak nuclear force. I mean, it's weaker than both of them. They do this side by side, buckler stars, atoms. So plot both a normal discipline on a long scale means separate by 2 log units. The neighbors on the axis, but not the same neighbors. Overlap only at cherry-picked edges. Uranium versus trans-Neptunian object. Very good. Completely false. No fancy physics needed. Calculator and radius table shows proportional centers don't overlap. Variability profiles don't match. Fixed orbiter versus variable orbiter. Just pick a high enough n is curve fitting, not prediction. Oh, claim 2. Damn, I like this. If you don't know that Caveman, it's a plugin that's basically uh, meant for LLMs not to vomit with a lot of text, but just speak super clean and clear. And so basically Caveman the result. Claim 1, dead. Claim 2, inverse square mass equivalence, still legitimate kernel of original intuition. What explore next? Claim 2, claim 1 dead. Claim 1 dead. Claim 2, the two body in this force problem production. I think claim 2 is dead as well. Category dead. Very good. Claim 4. Unface evaluable piece. A claim that nature is self-similar across atomic, stellar, and galactic scales is not currently testable as stated because no quantitative matching threshold is defined, no mechanism is proposed, and most developed formal version of this claim has been published since 1987 and not adopted by mainstream astrophysics. Interesting. I want to see what their claims are. Let's do a peer review, right? That's what we're doing. Oh my god. Hierarchical and fractal nature. The universe is nested hierarchy where fundamental system on one scale have analogous counterparts of other scales. Okay, now I know the name for it. By the way, is it a side quest? No, that's just definition. We didn't get brand new idea. So SQC still 3. I like this. I'm not saying that's true. I like the description of that. Scale transformation equation. Corresponding parameters such as the radius, r, time, t, and mass for system on adjacent scale are related by constant radius n equal kr, what is k? N minus 1. Time n equal the coefficient, right? Tn minus 1 and kd mni. Wait, k 5. Oh, they calculated it. 5.2 multiplied by 10 and 17 nd. I mean, they looks like they try to fit results into the theory, right? Core knowledges, stellar scale object Neutron stars are considerate analogies to atomic nuclei, and stellar planetary system are analogy to high Rydberg state atom. Prediction and success. The model successfully predicts the existence of stellar scale MACHO, massive compact hollow object with average mass of 0.5 m, pulsar planet system, and specific cutoffs in stellar mass function. Unlike the Big Bang model, this model is based on a fractal paradigm that's precedes an unbounded or least highly extensive discrete. How many complicated words you can put in one sentence? This is what it is. They lost me on a fractal paradigm that persists an unbounded or at least highly extensive discrete hierarchy of scales. I mean, when I read it a second time, it's totally makes sense. Aldershot models. Interesting.
Oldershaw.
Aldershot model. A fractal hierarchical cosmology proportions that the universe is organized in discrete self-similar scales with physics on atomic, cellular, and galactic level being analogies. That's interesting. Self-similar cosmological models. I'm gonna take it into my notepad.
Keplerian.
Did I really just speak on a mute for like 30 minutes? That's so bad. I don't know where I stopped, but I'm gonna repeat one thing that I think is important. So all this been given to LLM as a test to test the pipeline, and it's kind of succeeded in terms of set like everything is garbage, which is true, but it did not produce a proper scoring, and it did not drop that idea in the very beginning. Remember 15 out of 15 score? That should not happen. It should be 1 out of 15. Or the stage where it's dropped, it should be way earlier, but it didn't drop any stage. And what's interesting, we found out about LLM and the real pitfalls about this. The LLM couldn't find any literature about Aldershov SSCM to basically figure out how that number been calculated. And the problem with that's my assumption that in science and scientific community, there is some ideas produced that is so conceptually wrong and just not possible that majority of science, like 99% of them, don't even bother to recalculate or verify them or peer review them because it's just nonsense. And those kind of ideas are really, really bad, or those are magnets for LLMs pitfalls. They basically, why? Because everything published on the internet, right? Everything, like everything what's published on the internet has been read by LLM and feed the data. So LLMs go into that idea, read the idea, but it couldn't find any text, any validation that it's wrong, just because nobody bothered in saying it's wrong. And therefore, it identifies this as a real opportunity because nobody actually said this is stupid in a basic English, right? So we have to be very careful of those. If ideas so out of space, and I'm not saying novel science, it's just stuff that's, what word should I put it? Like, I'll give you example. What if we use yoga mats for lizard to improve their fitness? Like, what kind of idea is that, right? And then someone publish it. And I feel like it's going to be more and more stuff will be published like that just because of AI slap. The real scientists will not review it and will not put the effort and time into calculating some numbers just because its idea doesn't make sense, or concept, or claim. The claim doesn't make sense. And what's funny, when I was giving an example, I didn't even create any claim, so it was terrible example. But that example was to emphasize how stupid something can be sounds for real scientists. So those pitfalls for LLMs are legit. So and then I ask LLM like, hey, why you didn't drop that idea earlier? And that was its reply to me. Pipeline measure wrong things. Hey, here you go. So 15 out of 15 means agents did the assigned jobs well. It does not mean idea worth pursuing. Pipeline pass garbage idea with high marks because each agent executes tasks correctly. Sharpening split claims, scout find, counter evidence, red team kill it, synthesize stakeholders. That's a good process on a bad idea. This is a good process on a bad idea. Rubric rewards processing, ignore idea merits. The concrete gaps. No novelty gate. Idea has well-known name planetary atom model. Lineage to Aldershot self-similar cosmological model. Should after fail. Pipeline did surface this in stage 2 but did not wait it. No early drops verdict. Pipeline always run all 6 agents even when the stage 1 or 2 should kill the idea. Wasted tokens, diluted signal. No idea metrics dimension, impact effort. Probability nowhere in a rubric. Is it rubric?
Rubric.
Rubric. Your define metric. Propose fix architecture. Need 3 changes. Need triage state. Agent 0 runs first, can stop pipeline. Love it. New agent triage. Produce 0 triage MD before sharpen runs. Cheap 30 seconds. Web search. 1 pass. Coordinator. Novelty. Does the idea already have established name privilege? Possibility in principle. Love it. Traceability for self-learner. Love it. Hard drop rule if triage. Yes. For any atom galaxy ideator with score novelty 0, planet Aldershot, yeah. Same stuff. Caveman, please turn in and just give me the output. Add idea metric scoring to stage 6. 2 scores now, not 1. Merit access, yes. Change verdict. Implementation footprint. 1 things to configure before write it. To design decision need your call hard drop threshold. Propose any axis equals 0, hard drop or average. Alternatively, only novel hard drop. Confirm choices and I implement or push back. Confirmed. Confirm this. Should triage be allowed to fail ambiguously when triage agent uncertain, novelty unclear, found similar but not identical prior work? Should pipeline run with warning or should ambiguity itself be dropped? Confirmed. Drop when ambiguity and wait for user input to provide clarity into one or the other direction. In other words, start a dialogue and brainstorm session versus assumptions and research automatically with human. I have to pick up a daughter. I'll be back in 10 minutes. I'm back. This is classic. What happened? You basically set up a prompt, you give all direction, you think it's a job for, I don't know, 4 hours, 5 million tokens. You click enter and you go to bed thinking that you will wake up and everything will be ready. And you wake up to find the confirmation that happened like a few seconds after you left, and all that time and limits and tokens was actually wasted because not used, and you have no results in the morning, and you're like, leave that. And by the way, we finished this one. We finished reading the results. I'm surprised. 30 minutes speed run for 4 questions. Question 36 is waiting. What is the blood made of? Sorry. I think the blood made of many different cells that's produced in a bone marrow. Some cells produce in liver. The blood red cells has no mitochondria. That's what I remember. It's just because when you transport the oxygens, you don't want something to eat that oxygen, right? I also know there is a bunch of different cells and probably free-floating proteins, microelements from broken down stuff from the adjacent system that get sucked in through the adjacent system into the blood, and it's delivered to every single cell in our body. Pretty much a bunch of raw material, I would say amino acids or blocks of 3 or 4 amino acids together. So those amino acids could be delivered to the cells and used as building blocks for the proteins that's need or that's dictated by RNA. What else? Probably water in the blood. Maybe, maybe not. I'm assuming yes. There is some macrophages in other cells, the immune cells definitely floating there. Some antibodies floating there. Yeah, I actually would be very interested how many unique types of cells or microelements are in a regular human blood. And do the scientists know them all? Notice how I flip the tables? LLM asking me, and instead of answering, I ask them. By the way, clip that. For day 11, that's what I knew. And this is classical LM in our, like, what we just discussed. Is like, so, I forget the word again.
Sycophantic.
Sycophantic. So sycophantic. Really strong answer with some impressive details. Let me mark it up. So sycophantic. Producing bone marrow, correct. Red blood cells, white blood cells, and platelets are all made in bone marrow. Great. Some producing liver, correct for embryonic development. Interesting. The fetal liver is a major blood cell factory. Fetal, that's not a fetal. What's the name? Not the name, how to pronounce it?
Fetal.
Fetal, yeah. The fetal liver is a major blood cell factory before bone marrow takes over. I had no idea. Like, this is interesting. And when the bone marrow takes over and why it's take over, this is so cool. Like, imagine evolution build this system that's temporary until the bigger system take over and this one is obsolete or now serve for different purpose. Clip that. In adults, the liver produce many blood proteins. Albumin, clotting factors, but not the cells themselves. So half rate depending on interpretation. Red blood cells have no mitochondria, correct, and your reasoning for why is exactly the accepted explanation. Blah. Red blood cells also have no nucleus. They ejected during maturation to make more room for hemoglobin. Yeah, I forget that. I forget that there is a cell that's basically saying like, hey, no need for nuclear. I'm going kamikaze on this. Like, I'm not gonna divide, I'm not gonna reproduce, I'm not gonna be I'm serving one function. Load as much oxygen or hemoglobin as possible and run with that. They're basically bags of hemoglobin. This also means they can't divide or repair themselves, which is why they only last 120 days before being recycled. Allergy is killing me. Free-floating proteins. By the way, allergy well explained on a biological level, on a cellular level, and specifically with immune. Yesterday we learned that sterile mice who grew up without in sterile environment, without bacterial biome, they so acceptable to allergies because their immune system not developed. It's amateur. It doesn't know what to attack and not, and then any single like pollen landed in the nose, it's trigger like emergency signal versus like, oh no, it's just pollen, ignore it. Clip that. Next. Free floating proteins, correct. Oof, I was right. Blood plasma is full of proteins, albumin, antibodies, clotting factors, hormones, complement proteins, nutrition from the digestive system absorbed into the blood, correct. Amino acids, glucose, fatty acids, vitamins, minerals, all transported via blood to every cell. Glucose as well, fatty acids, vitamins, and minerals. By the way, what's the difference between vitamins and minerals? Minerals, I'm assuming it's just single chemical elements like potassium, right? Or ferrium, or like the iron, Fe, or maybe magnesium. But vitamins, it's more molecules. Let me ask this. That's gonna be a side quest. side quest count. But let's not do it here. The context here is full. I'm gonna open new chart. Where's my projects? Projects. What is the oh, the search that's now new projects. Is the difference between that could be just a simple Google search, but anyway, we have blood cells. Amino acids as building blocks delivered to the cell. Use per RNA instructions. Correct. You just described the full loop again. The adjacent absorption, blood transport, cellular uptake, protein synthesis via central dogma. Clean. Water, correct, and it's the majority. Blood plasma is about 92% of water. Macrophages and immune cells, correct. White blood cells, leukocytes, leukocytes,
Leukocytes.
Circulate in blood. Macrophages are slightly special. They're mostly in tissues. But the precursor monocytes travels in blood and become macrophages when they enter the tissue. Antibodies, correct. Free floating in plasma produced by B cells. The full picture of blood composition. Blood separates into two main fractions if you spin it in centrifuge. Plasma 55%, the liquid portion, water, proteins, albumin, antibodies, clotting factors, dissolved nutrients, glucose, amino acids, lipids, hormones, waste products, urea, CO2, and electrolytes, sodium, potassium, calcium. And then cellular portion, 45%. Red blood cells, erythrocytes, 95% of blood cells by volume. 5 million per microliter. Carry oxygen via hemoglobin. Hemoglobin is a protein. White blood cells, leukocytes, 5,000, 10,000 per microliter. Multiple types, neutrophils, most common first responder. Lymphocytes, B cells, and T cells, adaptive immunity. Monocytes become macrophages, eosinophil, basophils. Platelets, cell fragments, not whole cells that's clumped together to form blood cloth when you are cut. 250,000 per microliter. Your question, how many unique types are in the blood and do we know all of them? This is a great question with layered answer. The classic textbook list maybe 15-20 cell types, but modern single cell sequencing analyzing gene expression and individual cell has exploded that number. Blood contains hundreds of distinct cell subtypes and states. A single category like T cell now breaks down into dozens of subtypes: helper T cell, multiple subtypes, killer T cell, regulatory T cell, memory T cell, exhausted T cell, each with different gene expression profile. Okay, I can see it. Yep. For the non-cellular components, proteins, metabolites, lipid, microRNAs, the number is in the thousands. The blood proteome. So that's the plasma, right? That's what we're talking about, not cells.
proteome,
Proteome alone contains over 4,000 identified proteins. The metabolome adds 1,000 more small molecules. Scientists keep finding new ones, especially low abundance proteins that were previously below detection thresholds. I need one more break. Contains over 4,000 proteins that metabolize thousands of small molecules. Scientists keep finding more specifically. So we don't know them all. Every improvement in detection sensitivity reveals more. Blood is essentially a liquid biopsy of your entire body state. This is very important, and this statement could be legit reason to open a new biotech startup. And I believe, if I'm not wrong, that's exactly what that lady did, right? That's been posted in the Forbes. She promised 400 tests in a single drop of blood. So her premise being, I don't remember her premise, but he was saying that they invented technology that identify 400 biomarkers from a single drop of blood. And why everyone trying to do that? Because if you know the current state, and if you can access the current state easily, then you know the dynamics and trends. You can identify the issue way faster, way more simpler and more precise. Clip that. And this is the lady, fraudulent startup printed in Forbes. This startup raised 5.2 million to develop blood tests for cancer. No. Oh. I know this guy. Continue reading. Oh, come on. The paywall? I just need to find. That's probably here, right? Maybe not. I remember she was like dressing up as a Steve Jobs. Oh, this one. Elizabeth. The founder of the fraudulent blood testing startup, Theranos, famously styled herself after Steve Jobs, wearing black turtleneck, and was lauded by publications like Forbes as the next Steve Jobs. Before her company was exposed for fraud, she was convicted in 2022. Yep. That's the one. Is it sidetracked? Yeah, we'll call it as a sidetracked. 5. What am I reading? Almost every organ leaks traces of its activity into the blood, which is why blood tests can reveal so much about your health. 8 out of 10. Impressive, Breads. You hit red blood cells with the mitochondria detail, white blood cells, antibodies, nutrition proteins, and water. Missing platelets. I knew about them. I knew them because I didn't know how to pronounce them, and that's honest truth.
Platelets.
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 Platelets. Platelets. And the plasma cellular distinction, but overall picture was strong. What is the difference between dominant and recessive allele? Allele. I don't even like. Here's the story. Allele.
Allele.
That's the story how I came in United States, and on my interview to the as a software engineer for one of the companies. That's probably gonna be quick side. I mean, not a quick, the long side quest. But it's so funny because I was an interview. It was like 3 or 4 rounds of interview. And in the first round, I think it was 20 of us. Then in the second round, it was 5 or 4 left. I mean, selected, right? And then third round, it was only 2 of us. me and fresh graduate, and he was sitting for some reason we were in the same waiting room, which was so uncomfortable. And the last interview is supposed to be with the CTO itself of the bigger company, not the company that we were applying, because the company we were applying, they didn't have expertise to interview properly. So they brought a partner company CTO to basically vet at us and validate whom to hire. And that fresh grad was like good looking, like from the magazine, probably A+ student, nice suit, everything. And my situation was completely different. I could barely speak English, barely understand it. I was rough looking just because I didn't have enough, like not enough. I didn't have much money at that time. I was like living with a bunch of roommates in Coney Island projects. Like, have you seen the movie Requiem for a Dream? That's where I rent a place in a room or bed in a room for like $150 per month, like something crazy like that. And obviously, I didn't have a suit. That's how poor I was. And I was sitting in that waiting room, thinking like, okay, like why am I even sitting here? I have no chance. And then he went to the interview, left, like, speaking nicely, shaking hands and stuff. And then they called me and hello, blah, blah. And then they asked me a question. And like all blood drawn from my face right away, because it's not that I don't know the answer. That could be a problem, right? But I didn't understood the question. So it's not like I'm lacking the knowledge, which is bad on itself. I couldn't even understand what they asking me. So that was funny. I'm not gonna spoil what happened after, but that's a story for the next time. Clip that. I don't know why allele here, but what is the difference between dominant and recessive allele? I don't know what allele is, but I'm gonna improvise. So in cellular biology and microbiology, I don't know what's word allele mean. ALLELE. I don't know what does it mean. So I'm not fully understand the question, but I would guess that dominant and recessive, it's a conversation about gene. When gene combines or peaked, there is usually from mother and father, the feature that genome dictates to the certain cells, right, are actually related which one is dominant, which gene is dominant, and which one is recessive. And if one of them dominant and another recessive, dominant will be there. And I think the famous example, the color of the eyes, the blue eyes is recessive, the brown eyes is dominant. So if you get one dominant, one recessive, the result would be dominant. If you have two recessive, result would be recessive. And if you get two dominant, result would be dominant. Therefore, sometimes some anomaly from a first glance happen when children get some features that was not present in neither parent. to recessive combination. You didn't know the word, but you knew the entire concept. That's the pattern of this whole quiz. I mean, I love this. Like, it always find the way to give a compliment. Thank you. And LLL is simply a version of a gene. That's all the word means. The gene for eye color sits at a specific location on a specific chromosome, but that gene comes in different versions. One version might code for brown pigment, another for blue. Each version is an allele. Oh, the options. So, okay, I see. In the software, we would call it. Wow, I'm spacing right now. I couldn't bring a software analogy. I was thinking about it. You have two alleles for every gene, one for mom, one from dad, and they might be the same version of different versions. Thank you for very helpful token prediction next output. Version of different versions. Same version of different versions. So yeah, I'm not even go into side quests in this. Now let me score what you said. One from mother, one from father. Correct. Dominant shows over recessive. Correct. Dominant plus recessive. Dominant phenotype. Correct. Recessive plus recessive. Recessive phenotype. Correct. Dominant plus dominant. Dominant phenotype. Correct. Blue eyes recessive. Brown dominant. Correct. As simplified example, children can show features not visible in either parents. Correct. That's exactly right. Two brown-eyed parents who each carry one hidden recessive blue allele, brown, blue, have 25% chance of blue-eyed child, blue. The blue was always there, just masked. This confused people for centuries before Mendel figured it out. Mendel, state brother. The vocabulary to attach to what you already know. Homogeneous, two identical allele. Homogen, I feel like I'm learning like new language.
Homozygous.
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 Homozygous. Two identical. Okay.
Homozygous.
Homozygous. It's two identical
Allele.
allele. Clip that. Heterozygous. Two different allele. That means hetero different. Genotype, which allele you actually carry, the hidden code. Phenotype, what you actually look like, invisible result. So two brown-eyed parent can't both have genotype BB, hetero guy,
Homozygous.
heterozygous, B for brown, B for blue, phenotype brown, the child gets BB, homozygous recessive, phenotype blue, the genotype was hiding information. Yeah, thank you. One important caveat, eye color is actually controlled by multiple genes. So the simple dominant recessive model is an oversimplification, but as a teaching example, for the principle it works and you describe the logic perfectly. So why 08.5? The only thing you didn't have was the word allele itself. The entire conceptual framework was correct, including the sub. Yeah, okay. Oh, 57.2. What happened? And we're getting closer to 60. What is the difference between the nervous system and the endocrine system? I don't know what's endocrine system. Endocrine system, I would guess it's systems that throw hormones. I'm gonna guess right now fully. I don't know what endocrine system is, so I'm gonna guess that's endocrine system. That's a system that regulates processes or regulates like thermostats, and I believe that process called regulation homeostate. So I believe endocrine system, it's a homeostate for our body using hormones. So basically certain enzymes and proteins, because hormones are enzymes are proteins, probably enzymes, maybe both, thrown into the body. And then when bloodstream get enough of them, certain areas of our body react on them. Example would be growth hormone. Or another example would be adrenaline. The main difference between nervous system, it's also the similarities that they both exist to send information from central brain to pretty much every other cell or some cells on our body, right? But nervous system do it through the electrical signal and only utilizing neurons, versus endocrine system using hormones and the amount of proteins in bloodstream, not protein, amount of those hormones in bloodstream as a signal for the rest of the body to do certain things. So both for passing information, it's just different way. Nervous system fast, reliable, but probably couldn't reach certain parts. And endocrine system is more general, slower, and I believe for different, yeah, that's it. And again, that was the assumptions that endocrine system actually is that's actually systems that communicate with the hormones. Let's see what complements. Excellent reasoning from the word endocrine alone. Let me score it. Endocrine system regulates homeostasis using hormones. Damn! Look at that. Day 11. I'm dropping knowledge already. What's the ratio, right? What's the tally? I learned that word. That's its primary job. Hormones released into the bloodstream, correct. Endocrine literally means secrete within. Hormones are released into the blood, not through the ducts. What is ducts? Growth hormones and adrenaline are the example, correct. Both are hormones produced by endocrine glands. Both systems send information different methods, correct. This is the key insights. Nervous fast, electrical via neurons, correct. Endocrine slow, chemical via bloodstream, more general, correct. Hormosis are enzyme or protein, probably enzyme. Go back to question number 13. Hormosis are not enzyme. Different job. Enzymes catalyze reactions. Yes, but I thought that some hormones are actually enzymes to do that catalyzation. Hormones carry signals. Some hormones are proteins, insulin, growth hormone. Some are lipid derived. Okay, so some of them are proteins and some of them are lipid derived, derived. Testosterone, estrogen, cortisol. Some are modified amino acids, adrenaline, thyroid hormones, but none of them are enzymes. This is the same enzyme as the default gas pattern from earlier. That's a hard truth, and it's hard to take. But now I will remember. Clip that. Nervous system couldn't reach certain parts. Interesting instinct and partially right. The nervous system can reach almost everywhere physically, but it can't easily coordinate. A slow, body-wide, sustained change the way hormones can. Very interesting. Puberty, pregnancy, stress response over hours. This needs a endocrine system broadcast approach. Oh, because it's expensive. Of course. Communication through the neurons. Okay, that's why we have two systems of communication. Not because neurons couldn't reach, it's because of expensive. Communication through the neurons require way more ATPs than through the endocrine system. Let me do that. Let me ask it. I think I see what you're saying. I didn't read to the end. I'm right now at the part where you're saying that I would guess that neurons couldn't reach certain parts, and that was a guess. Obviously not true. But I was questioning why I was saying that, because I would question like why a organism needed two communication system or two types of information passing. And then I realized by reading this that the reason is, and please correct me if I'm wrong, I'm guessing right now again. The reason is because communicating through the neurons, it's way more expensive in terms of energy, because you constantly have to reset that action potential, and the amount of ATP you burn by communicating signal over neurons is way more expensive than just broadcasted body-wide in the bloodstream. So for the long-running processes like puberty, pregnancy, or stress response, adrenaline, it's way too expensive to trigger those neurons for 24/7 for multiple days. It's better to broadcast it like, hey, we are under stress or under pregnancy. Do this so far, right? Is it my assumptions are correct or wildly wrong?
Pituitary. 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 Pituitary.
The allergy is killing me. I was on a mute for another 10 minutes. We have to fix that. are still on deck. Why do cells die? Name any reasons you can think of. The cell die because it could be damaged, number one. It could not produce its own function anymore. Some processes went wrong. Might be even accumulation of garbage, so it couldn't dispose enough stuff, so it just died. Some cells live, like regenerate way faster, like gut cells, because they are located in an acid environment, or red blood cells, but they cannot replicate, so we need more of them. Interesting. Yeah, I would be very curious why do they die. But my main reasons are something went wrong, some mutation, or they couldn't, some organelles are broken. Like if all mitochondria die in cells, the cell will die. Not enough energy, cell will die. Not enough water, cell will die. But this is all the causes what will kind of kill the cell, right? Or T cell will kill a regular cell. And this is all the reason why cell die enforced by environment. But my main question is why the cell die by their own decision? Yeah, give me the list. I don't know the answer to this. Our score of 657 will drop. Damage, something went wrong, correct. Okay, your external causes are all correct. Damage, broken organelles, environment, stress, immune attack. Let me score those quickly, then get the question you're actually asking. Organelle fell, mitochondria dying, correct. Not enough for energy, water, correct. This is called necrosis. Death by injury or deprivation. The cell swell, bursts, spills its content, triggered inflammation. It's messy and unplanned. Now, why would a cell kill itself on purpose? This is called apoptosis, from Greek fallen off, like leaves from a tree. It's programmed, deliberate self-destruction. The cell dismantles itself from the inside, shrinks, chops its own DNA into fragments, packages its contents into neat little bundles that neighboring cells quickly absorb. No inflammation, no mess, clean suicide. I had no idea. I had no idea that our cells commit suicide. I knew they could die from external factors in a messy way, right? Triggering inflammation and macrophages, my favorite. But that they can commit a clean suicide, that's so neat. Clip that. Why would evolution build the self-destruct button? I mean, it's a smart way to do it, to build any system, but let's listen. Development. Sculpt in the body, your hands start as a solid paddle-shaped limbs in the embryo. The fingers form because the cells between the fingers kill themselves. Oh my god. Without apoptosis, you'd have webbed hands. Same process curves out the space in your brain, hollow out tubes These become blood vessels and separates your toes. Your body is sculptured by strategic cell death the way a statue is sculptured by removing marbles. I had no idea. This is so cool. This worth a tweet. Tweet this. Quality control, removing damaged cells before they become cancer. Yeah, that's we know that. When the cell detects that its own DNA is too damaged, to repair its trigger apoptosis. This is largely controlled by P53, the tumor suppressor protein P53 inspects the DNA, and if the damage is beyond repair, it activates the cell destruction things. Do we have P53 to detect HIV? Tweet that. And if the damage is beyond repair, is activates the self-destruct sequence. This kills one cell to protect the organism. Cancer happens when the system fails. Damaged cells that should have self-destructed instead keep dividing. Immune system training, removing self-reactive immune cells. Oh my God, this is so cool. This is so cool. I was asking a question like what Alan Watts was saying, like if you got and you control the everything, right? Who controls the gut? Like, and this is relatives to AI and who controls the guts, right? If you want to control everything, we or who control us. Any societies that have organization of control should ask the question who control those organs who enforce the control. Like this police, a classic example. And I was wondering, like, if immune cells, the T cells or killer cells, go nuts and start to killing its own body, I was wondering, classic autoimmune disease, right? I was wondering who controls the T cells, who will kill them, who will kill the killers, right? And this is exactly what it is. The clean suicide that's called apoptosis will actually eliminate the
Apoptosis.
Eliminate the killers. Your immune system general T cells with random receptors. Some of those receptors accidentally match your own body proteins. If those T cells were released, they'd attack your autoimmune disease right away. Here we go. So during development in the thymus, the T cells that react to self are forced to undergo apoptosis. Roughly 95% of developing T cells are killed this way. Only 5 that's correctly ignored. Self and recognized for region survive. Your immune system is built on the foundation of mass programmed death. This is interesting. So if they not kill themselves, then they replicate, I'm assuming, right, and they survive. But 95% of T cells are killed by, I forgot that name, self-determination, suicide. Of developing T cells are killed this way. Apoptosis. Infection containment. If a cell detects that it's been infected by a virus, it can trigger apoptosis. I mean, apoptosis, if cell detects it, the T cells will kill it, but I guess it will kill itself. To kill itself before the virus finishes replication, sacrifice one cell to prevent the virus from producing hundreds of copies and spreading. Some viruses like Epstein-Barr have a valve protein that specifically block apoptosis in the host cells, keeping the factory running. Interesting. Cellular agent, the high flick limit, 0.4, 57.2. I want to give this a little bit more thoughts and not rush and comprehend a little bit stronger or embrace this. So I gotta run right now to pick up my second daughter, but we will continue from this point next time. See you in day 12.